Here we go. Good afternoon, everyone. It's my honor and my pleasure to welcome you here today to a research talk in our seminar series here at Trinity College. I'd like to welcome the students, faculty, perhaps some staff members, and also some alums that are here to enjoy this talk today. And today we have the pleasure of hosting our visiting professor, Dr. Sally Seraphin. Dr. Seraphin is a primate behavioral neuroscientist who specializes in the evolutionary developmental neurobiology of stress. She has a BS in psychology with a concentration in biobehavioral studies from UMass Boston, a master's in human biology from Oxford University, and a PhD in anthropology from the Center for Behavioral Neuroscience at Emory University. In addition to this, she completed postdoctoral fellowship training in cellular and molecular neurobiology and developmental psychobiology at the Department of Psychology at Harvard Medical School and McLean Hospital. Today's presentation is inspired by her graduate research investigating the effects of early rearing on the behavioral pharmacology and neuroendocrinology of dopamine in rhesus monkeys. So we're going to enjoy a wonderful presentation by Professor Seraphin. And then after her presentation, we'll have some time for questions. And what I wanted to offer is for if anyone has any questions they think of during the talk, feel free to type them in the chat. And I can try and moderate and field those questions, but it's often nice to kind of accumulate some questions as the talk goes along. So I will try and continue to monitor the waiting room and let people in. But other than that, Dr. Seraphin, welcome.
Thank you so much. I'm really excited. Oh, you've disabled screen sharing. So
sorry about that.
Oh, it's okay. I'm really excited about this talk, and I've planned to talk about a lot. So I hope you've got an appetite to hear what all I have to say. And can you see the slide, the title slide, or can you see my notes? Which do you see?
I see the title slide. It looks great.
Okay, fantastic. So that's what I want you to see. And I will tell you that I, even having taught online for a while now, under the pandemic, find it difficult to monitor your faces during the delivery of the presentation. So please forgive me if you have an emoji or a hand raised and I don't see that immediately. If you can reserve questions to the end, unless there's something really pressing, please do so, and then we can have hopefully a hearty discussion after the talk. So my goals today, and here you see the title of the presentation, Human Experience Through the Lens of Dopamine, are first to help students. I have a lot of my undergraduates here attending this seminar to help you students of neuroscience become more holistically acquainted with the properties of dopamine. And then my secondary goal is to prime my colleagues, really, those more seasoned neuroscientists, including the alumna among us, for future discussions on the benefits of a more socio-ecological or neuroethological approach that roots neurobiology in richer evolutionary context. So forgive me, I have two screens going, and I will bounce back and forth between the screens. So the first question is essentially, what is dopamine, right? And this is really probably at the forefront of your minds today. What exactly is dopamine? That's going to be the first thing that we address in this presentation. And here's some information about what dopamine is. You can see the organic structure, 8 carbons, 11 hydrogen molecules or hydrogen atoms, 1 nitrogen. And there you have it, dopamine. The important thing to note about dopamine is that it's one of the more important neurotransmitters. It is one of the monoamines, as you can see from the illustration in the upper left. All monoamine neurotransmitters contain an amine group. A subset of these that we call catecholamines also contain a catechol chemical group. And that basically is what defines dopamine. It's one of the monoamines and more specifically a catecholamine. So dopaminergic neurons produce dopamine. They are located in specific parts of the brain, and we'll talk about where those parts are, but their projections go everywhere. And that's something that's critically important for my argument today about the omnipotence, really, of dopamine. So you can see that, and hopefully you can see my cursor movements, that tyrosine is a starting point, and tyrosine hydroxylase turns tyrosine into L-DOPA, and then we have another enzyme that intervenes and converts this to dopamine. Further down the biosynthetic pathway, we get noradrenaline and epinephrine, which we won't be talking about today. So where is dopamine is that next part of the discussion. And dopamine is everywhere because it has such far-reaching axonal projections. In their book, The Molecule of More, Lieberman and Long describe serotonin, which is related to mood, oxytocin, which you've probably heard referred to as the cuddle chemical, related to social bonding and attachment, and endorphins, which are related to the feeling of a runner's high, for instance, and has the capacity to, soothe feelings of physical pain. They refer to those other three chemicals as here and now chemicals that relate to the modulation of internal states, or what they refer to as parapersonal spaces. Dopamine, by contrast, is a source of both creativity and madness in that it relates to extrapersonal spaces. It helps us navigate that divide between ourselves and the world around us. It helps us to look up and out instead of down and in. And therefore, depending on whether you have an internal or an external locus of control, it can be your friend or your enemy, right? But in any event, more on where is dopamine. There are four brain dopamine pathways. There's the mesolimbic pathway, the mesoprefrontal pathway, the nigrostriatal pathway. And humans have about 400,000 to 600,000 dopaminergic neurons. About 70% of these are located in the substantia nigra pars compacta, which is where they begin, actually. And so you can see the mesolimbic pathway, labeled pathway 1 here, and I'm sorry that the resolution looks a little bit fuzzy on your end, I think. Goes from the ventral tegmental area to the nucleus accumbens. That second pathway goes from the ventral tegmental area to the prefrontal cortex. The third pathway goes from the substantia nigra to the striatum. And the fourth goes from the infundibular nucleus of the thalamus to the pituitary. So we'll talk about these in more depth. The important thing to know is that dopaminergic neurons originate in the midbrain, and then they radiate throughout the brain where they support all of our beautiful human functions. So the other piece about dopamine that's worth thinking about is that it's dopamine genes correspond not only to this protein, this neurotransmitter rather that we make, but also to all the members and an assortment of characters really that support dopaminergic neurotransmission, one of which is the dopamine transporter. Can you guys see this illustration of the dopamine transporter? Yes?
Yes.
Okay, fantastic. Thank you. So the dopamine transporter functions like a vacuum cleaner. So whenever the presynaptic neurons pumps out some dopamine, depending on how much of that dopamine is available, if it's bound to the receptors or not bound to the receptors, anything that's in excess or that has served its purposes is sucked back up by the dopamine transmitter. Therefore, it has the effect of limiting the stimulatory activity of dopaminergic neurons by reuptaking essentially any previously secreted dopamine. Cocaine, for instance, many of you think of dopamine and remember cocaine and addiction, blocks dopamine transporter, allowing dopamine in much the same way that SSRIs keep serotonin active in the synaptic cleft to promote the mood enhancing properties and counteract depression. Cocaine keeps dopamine available, blocking those dopamine transporters, allowing dopamine to interact with its receptors repeatedly. And therefore you get cocaine intoxication and associated feelings, again, in keeping with that idea of madness, of increased energy, euphoria, racing thoughts, enhanced motivation, particularly to pursue set goals that really are hard to distinguish from states of mania. So the other key players include the receptors, right? So we have dopamine, it has receptors and it has this transporter that mediates its access and availability to receptors. So dopamine receptors fall into two main categories. They are class 1 or class 2 receptors. Dopamine receptor 1 receptors excite adenylate cyclase activity, whereas dopamine receptor 2 receptors inhibit adenylate cyclase. So those of you who've studied some biochemistry will know that adenylate cyclase is the enzyme that synthesizes cyclic adenosine monophosphate or cyclic AMP from adenosine triphosphate. So literally these two subtypes bind dopamine, and through them dopamine is able to transduce signals that essentially impact, the functions of neurons importantly distributed in a variety of brain regions. So you can see from the top illustration on the upper right-hand corner here that dopamine is the key. It binds these 5 different classes of receptors that have some overlapping, but some unique distribution patterns throughout the brain. You can also see that there has been some genetics done on these receptors, and we know which chromosomes they belong to, the extent to which they have introns, how many exons, and so forth. This is particularly important information as it relates to our later conversation around particular polymorphisms, particular alleles of dopamine receptors that relate to aspects of personality and even large scale cultural and evolutionary changes. So like politics, all dopamine is local. So they have overlapping, but still quite different distributions. You can see that the D1-like receptors, dopamine receptor 1 and 5, are located in the caudate, the putamen, the nucleus accumbens, the hypothalamus, thalamus, frontal cortex, and that the D2-like receptors have a slightly overlapping but different receptor distribution. The important and interesting thing to note is that the dopamine receptor 4 is especially localized in the amygdala. We'll talk about that momentarily also. So what does dopamine do? So it's in lots of different places, and it is important for looking up and out instead of in. So what does it do actually in the brain? Maybe your next question. So at this point, I'm going to go through these 4 dopamine pathways and bring in different kinds of evidence to build this idea of a holistic, sort of widespread importance of dopamine in terms of human behavior and human experience. So you'll remember me mentioning that first pathway, the mesolimbic dopamine pathway. So it's involved in processes of addiction. It is important for telling the organism something about salience, really what environmental stimuli or variables deserve or warrant attention. It originates in the ventral tegmental area, projects to the shell of the nucleus accumbens, the amygdala, the hippocampus, and the olfactory cortex. All of these are parts of the limbic system, which mediates or helps control or modulate, depending on your word preference, our emotional lives. So in keeping with this, it's probably no surprise to you that mesolimbic dopamine is involved with motivation. And what that means is that it is involved in the hedonistic properties of our existence, our pleasure seeking and our pain avoiding propensities. For decades, psychologists have viewed the neurotransmitter dopamine as a double-edged sword because it is released in the brain as a reward to train us to seek out pleasurable experiences, but the pursuit of those pleasurable experiences can lead to devastating results, right? And before we dive into a discussion of addiction, I want to sort of calm things down a little bit by, not that you're all overheated, but maybe I am, I don't know, by talking about appetitive behavior. So appetitive behavior has to do with our not only sensation seeking, but our seeking of nourishment and social interaction, all those things that map onto our basic needs in a sense. Dopamine, when it comes to eating, which is the ultimate appetitive behavior, right? The word appetite's in it, is released twice. According to this recent article in Nature in Neuroscience Nutrition, excuse me, our brain rewards us for every meal by releasing dopamine, not only at the time that the food is ingested, but also once it reaches the stomach. So we are, our attention to the essentials for maintaining life, essentially, are modulated by dopamine and its rewarding properties. And so it is the case that with addiction, unfortunately, this process of pairing behavior to stimuli in a way that maximizes reward becomes completely unhinged. The recent and still unrolling ongoing opioid epidemic is a testament to that. still ravaging formerly industrial towns throughout Appalachia, the Midwest, and, former mill and fishing towns in New England. These diseases of despair, essentially, that are engendered by economic devastation, and perpetuated through structural inequalities, essentially create fertile ground for the rooting of addiction, this wanting, this searching for something to alleviate psychic pain that people sometimes experience with economic devastation and structural inequality. And so, you may not be addicted to food. You hopefully are not addicted to drugs. You can see that not only does dopamine interact with centers of the brain, I'm sorry, do drugs interact with centers of the brain wherein dopamine receptor 2 are active, essentially reducing the densities of those receptors over time by constant use. But there's increasing evidence that our smartphones and those devices that specifically administer variable reward that leads to increased reward prediction errors. So those of you who've taken cognitive psychology will probably have heard about this, but it's reward prediction error that is specifically involved in our desire and our constant attention to those mobile devices that periodically will give us an unexpected source of pleasure. That text from a friend you hadn't heard from, a really funny meme, a like on Facebook, all those things cause subtle release of dopamine that is enchanting and ultimately a source of addiction. So the next pathway I want to talk about is the attention pathway. And you've probably heard this invoked, the mesoprefrontal dopamine pathway, in the context of disorders like schizophrenia, attention deficit hyperactivity disorder, or ADD, as well as autism. And the important thing to know about this pathway is that it regulates working memory by activating neurons in the prefrontal cortex, which is a domain of learning, attention, executive function, short-term memory, planning, and perception. So hopefully your awareness now of dopamine and its widespread influences is building. It's important to note that this attention pathway also spans or reaches into the hippocampus, which maintains a number of different kinds of memory, including declarative, spatial, and contextual memories, and functions also in the detection and comprehension of novel stimuli, which is especially critical when it comes to human evolution, as we'll get to before long. So now I'm going to do a sort of quick and dirty tour of some aspects of cognition that are modulated by dopamine. These two illustrations are from a 2016 study that showed that mesoprefrontal dopamine regulates many aspects of cognition, including attention, as I mentioned before. The same study showed that sleep deprivation disrupts dopamine signaling and impairs attention. So I want you to remember this piece about sleep deprivation and its impact on the disruption of dopamine neurotransmission. The other study I want to show you is from 2009. And this busy slide is essentially here to tell you that mesoprefrontal dopamine regulates aspects of cognition, as I mentioned before, but that it's especially involved in difficulties in ADHD, where there are reductions in the availability of dopamine receptors, as well as dopamine transporter in association with the attention deficits experienced by people with ADHD. So dopamine also relates to personality. As I mentioned, that book that describes it as helping us to look up and out sort of telegraphs this notion that is supported by evidence, in fact, that extroverts, people who look up and out, particularly for social interaction, tend to have be more interested, hyper attuned, if you will, to dopamine, that they, people who are measured as extroverted based on the 5-factor model, OCEAN, have been studied, and data shows that in comparison to introverts, they are much more sensitive to dopamine, with introverts, being more sensitive and less interested in having excessive dopaminergic stimulation. And this idea about personality as it relates to introversion versus extroversion. And now you see a slide with Fisher, Lee, and Verzijden, right?
Yes.
Yes. Okay, fantastic. Sorry about that. So in the previous slide, what I wanted you to take away is that extroverts require larger amounts of dopamine, whereas introverts are highly sensitive. In this slide, what you should take away is that there are cross-national differences in personality that also relate to our sensitivity to dopamine. And in particular, this tendency to like dopamine, just, in terms of the shorthand, wanting more, varies internationally. And it varies in a way that maps on with some broad cultural attributes related to extroversion and introversion. And these gene by environment interactions on personality are really difficult to tease apart because of all the factors involved. Also, people migrate and people, define themselves on the basis of many factors, for instance, many cultural factors. But there's one theory, at least, and that is the theory that Fisher and colleagues describe in this 2018 study, that suggests that climatic stress, that genetic differences in dopamine that are predicted by personality traits have something to do with living in a more or less demanding climate, particularly one that is stressful in terms of access to resources, or stressful in terms of exposure to diseases, for instance, such as parasites. So this particular approach calls to mind a psychoneuroimmunology of dopamine, the idea that our genetic differences in dopamine may relate to living in climates that expose us to varying degrees of sickness and disease. The other sort of cultural approach that I want to mention that relates to personality, and maps onto this idea of extroversion and introversion, is one by my collaborator, Shinobu Kitayama, and his colleagues. So they've done a series of beautiful studies looking at specifically dopamine receptor 4 and brain morphometry in people of Asian descent and white Americans. And essentially what they've found is that people of Asian descent, and this is in keeping with our notions about what sort of life might be like in a society like China, or what we can observe in studies of emotional tendencies in Japan, that people from societies that are collectivistic define themselves very much in relation to other people. And what you see is that they also score higher in measures of interdependence or self-construal that's interdependent. By contrast, people who come from Western European heritage in the US, in particular, define themselves more independently, and their self-control, the way that they perceive themselves and what's important in terms of social interactions, is much more independent and less interdependent. And this is even more so when people are carriers of a particular polymorphism of DRD4 they have found. And then above and beyond that, there is some differences in terms of the size, the volume of a particular part of the brain called the TPJ, or the temporal parietal junction, that correlates with more interdependent self-construal in carriers of the DRD4 gene. So I hope this is really interesting to you, because obviously it is to me, but this idea that somehow there's been this gene culture coevolution really quite recently that has shaped the minds of people to match their particular social cultural contexts by virtue of dopamine's role. So what is the role of dopamine in human evolution? You're probably wondering. And we're going to talk about that. We're going to talk about it first by introducing you, I think, to some terms that you've probably never heard of. I'll be interested to know whether you've heard of Homo erectus, for instance. So one of the interesting things about dopamine, and this is, getting into a lot of theory, is that it is very likely to have been involved in the processes of human evolution. And by that, I mean, our becoming more upright, our increased encephalization, having larger brains, and all the beautiful things that humans do essentially have increased in prevalence, largely in relation to some of the functions that I previously described with respect to dopamine. So you can see here, and everybody can see this evolutionary tree with all the skulls, I hope.
Yes.
Yes, fantastic. So you can see here on the right-hand side, chimpanzees and humans share like 99% of our genes. And a lot of the variation seems to be in a very small percentage that dictates the pattern of gene expression. But one of the few gene systems that has shown an evolutionary departure between humans and our chimpanzee cousins is particularly dopamine receptor 5, but to some extent other dopamine system genes. So chimps are our cousins, and these are all the human ancestors. And you can see that we separated from chimps. We shared a common ancestor about 8 million years ago. With Homo habilis, about 5 million years ago is where we really started to grow bigger brains. We started standing upright and consequently spending more time looking face to face and developing some of the aspects of the grip that made it possible to create stone tools and manipulate fire, which is essential for processing processing meat for increasing its digestibility through cooking. So this happened with Homo habilis, and it really took off with Homo erectus. And with Homo erectus, we had some additional changes that are particularly important when it comes to the properties of dopamine. So meat eating is an essential component. Meat eating allowed Homo erectus to build body fat. And you've probably heard about the expensive tissue hypothesis, this idea, which is fact, that the brain is extremely calorically, intensive, second only to the gut. With meat eating, not only can you reduce the length of your digestive tract, minimizing the amount of calories needed to process food and maintain the workings of your digestive system, but you can also divert precious resources to building a larger brain. The other benefit of meat eating is that it's a source of tyrosine. And you'll remember from early on in this lecture when I talked about how tyrosine is converted to the dopamine precursor, levodopa. That has the effect of enhancing dopamine gene expression. More tyrosine is also associated with more glial cell-derived neurotrophins, which are basically chemicals that protect dopaminergic neurons, right? So we're growing a bigger brain that potentially has more dopaminergic neurons. And so this meat eating is a critically important part that relates also to changing ecological pressures. So with Homo erectus, we saw an expansion of territories, increased group size. So people's ancestors, hominids, started living in larger groups. They started pair bonding. One of the ideas about language is that it correlates with increases in group size because that enables us to socially groom and socially manipulate over some distance as group size increases. Homo erectus also developed handedness, and we know this based on an examination of tools and their uneven construction and wear patterns. And that means that lateralization has started to happen. So somewhere between Paranthropus and Homo habilis, people's vocal apparatus became disjoined from our breathing apparatus. And so it became possible for us to articulate words and speech. And so this lateralization may correlate to that as well, but based on the increased allocation of brain real estate to right hemispheric functions, I would argue that a lot of what we have come to accomplish is because of the strengths of, dopamine manipulating our interpersonal and upward and outward looking potential. So sleep changes are another aspect of increasing dopamine expansion, as is enhanced visual and spatial functions, including working memory. But the thing about sleep is that Homo erectus went from sleeping in the trees to sleeping on the ground. And it's thought that that shift enabled us to engage in longer bouts of integrated sleep, allowing for more REM sleep or rapid eye movement sleep, which is associated not only with dreaming, but is highly dopaminergic and associated with creativity, innovation, also the consolidation of procedural motor memories that you might need to sort of, shape your learning of how to build tools, and also visual spatial memory that is important for monitoring your territorial holding and defending that territory. So the other thing that we see in Homo erectus is that they become sort of better capable at traveling over long distances, not only because of their skeletal-motor, sort of anatomy, but, evidence suggests essentially that they, by virtue of this longer distance travel, that they would have been better at modulating body temperature, essentially all these hypothalamic functions that are associated, in addition to sleep and circadian rhythm, with temperature modulation or temperature homeostasis. We know that they were really incentivized in some way by, or rather to maintain certain territorial boundaries. And these are the first human ancestors to sort of expand from an original location to colonize other parts, other regions, largely in Africa. And so we can start to speculate about mesocortical activity rooted in dopamine among these ancestral hominids. And some of what, is theoretical has some grounding in what we have been able to observe, or can be rooted in what we have been able to observe with regard to dopamine neurotransmission and gene systems in migrating modern humans. So in the last 10,000 years or so, people, modern humans, have colonized, that's the word I'm looking for, every part of the world. And what we see is that these dopamine receptor 4 genes, and particularly the 7R allele, that same allele that was, connected to extroversion, is connected to novelty seeking and openness to experience, has been found to associate with our expansion and migratory patterns, with the prevalence of that allele increasing by 4.3% for every 1,000 miles that we humans migrated along this historical trajectory, leading out of sub-Saharan Africa into Asia, back into Europe, across North America, and into South America. So as you would predict, the highest levels, incidence of this DRD4 7R allele is in South Americans today. You can't talk about dopamine without mentioning sexuality. The DRD4, that same novelty-seeking polymorphism, has been found to associate with a number of phenomena, behavioral phenotypes, including ADHD pathology, alcoholism, tobacco craving, financial risk-taking in males, and other personality traits in keeping with the novelty seeking, but also in terms of sexual experimentation and the age of first intercourse are highly predicted based on the prevalence of this particular polymorphism. So I want to very quickly get to mention a couple of other dopamine pathways. The nigrostriatal pathway is important for motor functions. You'll probably recall, at least from the abstract of this presentation, that it's involved in Parkinson's disease and extrapyramidal disorders. Indeed, in the case of Parkinson's, we know that the tremors, the rigidity are examples of things that can be treated with the administration of dopamine agonists. It's also the case that the same brain regions that are involved in the modulation of movement, the striatum in particular, are involved in our feelings of elation, the feeling of chills that you get when you experience peak pleasure from the arts, so from music, for instance. And this is, you can actually observe the enjoyment, the process of enjoying music using PET and fMRI, and actually differentiate the period of anticipation from the period of peak emotion based on physiological measures, um, based on the striatal dopaminergic activation. So the striatum, again, being part of that nigrostriatal pathway that's involved with Parkinson's disease, but also supports our social desirability and our pleasure or enjoyment of music. So this particular illustration or slide, and you guys can see the slide that says social desirability, and there's a PET scan on the left-hand side.
Yes.
Right? Okay, thank you. So I'm including this slide just to mention that social desirability, thinking that friends and relations, seek your affiliation, is inversely correlated with binding of raclopride, which is a dopamine receptor 2 antagonist, in the striatum as well as in the hippocampus. And raclopride, so raclopride sits and occupies dopamine receptor 2, making it inaccessible to circulating dopamine. And so what this means is that more raclopride binding occurs in the striatum and in the hippocampus among those individuals that have rated slightly higher social desirability. Dopamine is also involved in the processes of lying, as you can see from this illustration, where again, raclopride, used as a dopamine receptor 2 antagonist, is associated with lying and social deception. So dopamine is not only influencing our experience of pleasure, but it's also involved in our Machiavellian intelligence in modulating our interactions with members of our species in a way that telegraphs social cognition and in particular social competition. A lot of studies have shown in the last 10 or 15 years that social status, the acquisition of dominant social status, is correlated with increased dopamine receptor 2 binding in various species of macaques or monkeys. Here you see crab monkeys and Japanese macaques just chilling in a hot spring over there, but also in studies of rodents. And in particular, in keeping with this idea of social cognition and these earlier studies that showed elevated dopamine in the basal ganglia subsequent to the achievement of social dominance, it's known that dopamine in the ventral tegmental area and substantia nigra, those points of origin of the 4 pathways, along with a recently discovered neuropeptide BW and its receptor, are also involved in the perception of social dominance. So dopamine not only helps us think about our status, but it helps us to think and monitor our status in relation to that of other individuals. My favorite dopamine pathway is the little discussed tuberoinfundibular pathway. And The tuberoinfundibular pathway overlaps primarily with the hypothalamus and pituitary. It's not considered as behaviorally important as the other three, but at the same time, as those of you who've studied neurobiology know, the hypothalamus and the pituitary are ground zero for a lot of our competitive social and reproductive functions, as well as our ability to modulate our stress response. So how is dopamine involved in epigenetic processes of development? And we're very quickly running out of time. I'm going to actually just very briefly tell you about some work that I've done in monkeys. And basically, a lot of these features of dopamine that we talked about before, including its involvement in social dominance and addiction and attention, also are seen in animals that have been exposed to early deprivation or early adversity. And some of the behavioral consequences of early maltreatment map onto the functions of dopamine just described. So I did a series of studies in rhesus monkeys looking at how the neuroendocrinology, the behavioral pharmacology of dopamine can vary in conjunction with maternal deprivation and peer rearing, or normal rearing with a mother followed by co-housing with a similarly reared peer. And over the course of a series of, almost 10 experiments, I was able to show that essentially one of the things that happens with early maltreatment is that your dopamine neurotransmission is impaired. So given raclopride, that dopamine receptor 2 antagonist that I mentioned before, rhesus monkeys that have been deprived of their mothers have less dopaminergic tone, which is measured by the prolactin response to raclopride challenge. And that's a discussion that we can have exactly how that works. Their neurochemicals also become sort of scatterbrained. They're no longer correlated in meaningful ways with one another as you would find in a normally reared peer. They are also differently motivated and influenced by drugs that interact with dopamine receptor 2 and 1 as agonists and antagonists. And let's see if I can get to the next slide here. They also show a loss of specific dopamine receptors in critically important regions of the brain. So this illustration just shows you the pathway, how you might administer raclopride and get increased prolactin based on raclopride's engagement with that tuberoinfundibular pathway that I so like. And this is the data. Mother reared have a robust prolactin response. Nursery reared, kind of pitiful. So they have lower dopaminergic tone. Indirect consequence to their their neglect, or this model neglect, in keeping with their lack of access to a stable caregiver. And I'm just going to fast forward and show you in the basal ganglia, we didn't see any differences with respect to dopamine receptor 1 or 2. But then when we looked in the prefrontal cortex, we saw significantly increased binding of dopamine receptor 1 in the orbital prefrontal cortex, which is associated with reward, the processing of reward, and in particular, the estimation of reward value. In the mother-reared, but not the nursery-reared, this would be consistent with the idea that nursery rearing disrupts one's ability ability to look upward and outward in a meaningful way and learn all those contingencies that can maximize your individual reward, your success in a complex social environment. And it was also the case that dopamine receptor 2 levels were higher in mother-reared animals compared to nursery-reared animals. So they, in essence, we would speculate based on the corresponding behavioral data, have better impulse control and are less likely, for instance, to challenge individuals that are socially dominant or bigger to fights and suffer sort of mortal consequences in the wild, in semi-captive environments. We see that nursery-reared animals that are models of early neglect and maltreatment, are more likely to take death-defying leaps from branches by virtue of their wanting impulse control, perhaps because of disruptions to dopamine receptor 2 activity in the medial prefrontal cortex. So the general conclusions, and that's me a few years younger, from the series of studies on rhesus monkeys were essentially that mother rearing shapes the coordination of those neurochemical systems. Not just catecholamines, but all the monoamines are correlated in mother reared, but not in nursery reared. Nursery rearing also leads to reduced dopaminergic tone based on their pitiful prolactin response to that raclopride challenge. Nursery rearing also changes the relationships between behavior and dopamine metabolites. That's data that I can talk to you about after the talk with anyone who's interested. And that it reduces prefrontal dopamine receptors that are important for doing the work of social cognition and motivation and, sort of memory. And also that it changes our behavioral sensitivity to drugs that modulate the effects of or impact the functioning of dopamine receptors 1 and 2. So what does a socio-ecological approach to dopamine look like? Really, what I think that looks like is an approach that doesn't overly dwell on the disease model, the Parkinson's, the ADHD, the addiction, but rather makes room for the rich social context and phylogenetic history that has made us what we are, individuals that not only look in and down, but upward and outward, and dream and aspire. Dopamine is critically important, really, for enabling humans to imagine and seek what never before has been acquired, experienced, and ascertained. And so essentially, that I think is what a socio-ecological approach to dopamine would look like. And I want to end here thanking you on the dot. I finished at 3 o'clock. I kind of had to rush at the end, but I am really happy to take any questions you might have.
Wonderful. Thank you so much. Fascinating tour through development, evolution, dopamine, the world.
Is your head spinning?
Well, there's so many things to kind of tie together and think about, which, it's exciting. That's what neuroscience is very interdisciplinary, to try and think about how to pull different disciplines together. I noticed that we do have a couple of questions in the chat.
I'm going to stop sharing the screen.
Stop sharing.
So,
yeah, then we can
yes.
see everyone, which would be great.
Yes. Okay, let's see if I can stop share. There we go. Okay. So in the chat.
So in the chat, I'll just read the second one from Barry Sheckley. Some descriptions identify the anterior cingulate and related network of spider neurons as a crap detector of deviations between expectations and the perceptions of a new situation. does dopamine play a role in this? And in rewarding accurate predictions for, and in a role in this crap detection, and in rewarding accurate predictions for a new situation and not rewarding inaccurate predictions?
So my sense from that literature is that the reward prediction error is more important than reward prediction successes. So if you do something that you know will get you dopamine, get you a hit, you get a little bit of dopamine. If you are sort of, shooting in the dark and you make the correct move, you get a massive dose of dopamine. So and this is part of the problem, particularly with early maltreatment. Is that it's like putting gas on the fire of impulsivity. They don't take a particularly predictable strategy, but they're constantly behaviorally chaotic and disorganized. That impulsivity overlaps with the reward seeking. They are hyper attuned to taking chances that maximize exposure to reward, big reward, in terms of that disconnect between, a poorly predicted reward and the actual acquisition of, hitting the jackpot, essentially. So I don't know if that answers the question. Barry, does anybody else have?
Yeah, we can keep moving. There's a couple of
Okay.
Oh yeah, here he is. There's a couple of other questions as well. Was that an adequate answer to that question? Oh, I think I have you stuck. Good. All right.
Okay.
Another question that came up, and these are two kind of probably fairly simple questions. Is there a relationship between levels of dopamine and diagnoses of ADHD? That's one question.
Mm-hmm. So you want me to take that before,
Sure.
or do you want to go on to the next one? Okay, so when we talk about levels, there are a bunch of different things that we can measure, right? So there's the amount of dopamine that's actually circulating, the amount of the catecholamine. Often in cerebral spinal fluid, we might only be able to measure its metabolite. So in those monkey studies, I looked at a lot of dopamine metabolites, predominantly homovanillic acid, as a measure of circulating dopamine. But with ADHD, my sense is that people are usually talking about the availability of receptors and or neuronal connections to support the kinds of complicated processes involved with attention modulation. So by giving someone a stimulant, you can increase the activity of whatever neurons are there to do the work, and you can increase the availability of some agonist to interact with perhaps what might be a limited population of receptors.
Very good. Another question kind of like that, is dopamine released during exercise, like endorphin, as an addiction creating reward?
Yes, so not only is dopamine released during exercise, but specifically dopamine receptor 2 seems to be connected to this leap by Homo erectus, from being not stationary like we are, but being almost seasonally migratory and encompassing a wide range. It's thought through its actions in the hypothalamus that it modulates your thermoregulation ability. So it enables you to run or walk longer distances without overheating.
Mm-hmm.
So it on the one hand has the benefit boosting our experience of the world subsequent to exercise, but it actually supports the metabolic functions that keep your body balanced during especially extended exercise.
Fascinating. Okay, another question. This is more about early trauma. In your opinion, how permanent are the effects of early maltreatment on the dopamine systems? Do these changes occur on an epigenetic level? And what would you suggest for repairing damage to the dopaminergic system that was caused by trauma?
So it is an epigenetic process. And this is one of the reasons why a socioecological, a neuroethological approach is important, because one of the things that we know is that a faster means by which genetic inheritance can unfold, or plasticity can take root, is through the direct transmission of patterns of gene expression beginning in the womb, through selective methylation and phosphorylation of genes. But also, it's the case that early in life, just as there are critical periods for developing sight, um, our patterns of our patterns of apoptosis, how your brain sort of is shaped and modeled based on the targeted death of superfluous neurons, our patterns of synaptogenesis, how your brain is wired, are critically dependent on input from the environment. So we are experience expectant, meaning that we expect there to be a stable caregiver who's a lot older, after which we can psychobiologically attune our body. And it's also experience dependent, meaning that if you get a lot of crap, to use Barry's word from earlier, you end up creating an individual who is particularly tailored for a crappy niche. And that makes it really difficult, not only to learn other habits that perhaps confer greater well-being, but it also is a source of opportunity. If we are able to recognize early childhood adverse experiences for what they are in a timely way, we can set up a whole host of interventions that can disrupt the negative cascade of phenomena, including earlier sexual maturity, earlier age of parenting, combined with a sort of that delayed frontal cortical development that doesn't even manifest until the late teens, early 20s in normal adults. So you have a situation where the individual who experiences early insult is burning a candle at both ends. There's evidence that there's advanced aging or advanced senescence, not only an accelerated sexual maturation, but also advanced aging based on the length of telomeres, the basically bubble wrap at the end of your chromosomes, in consequence to stress. So you burn out more quickly, you bolt through life, and yet these are all circumstances that could be interrupted with better early childhood education, with better supports to families, with young children, with more social and educational interventions, as well as nutritional interventions early on.
Yeah, and that might relate to another question. What about people who are able to succeed through discipline and hard work despite early trauma? So probably a bunch of factors that might.
Yes. So that is really an interesting phenomenon. And there's some monkey studies actually that kind of show something interesting, whereby if you breed matriline's, so monkey lineages, for punishing and neglectful physically harmful parenting, and then you cross foster the young from those harsh parenting lineages where they're stepping on their infants' heads and, knocking them into rocks, and, they're getting bumped and bruised and neglected. If you cross foster infant monkeys from those matrilines to matrilines that are nurturing and attentive, one of the things that you see is not only is the inherited effect, whatever vulnerability that may be genetically based and involving neurotransmission of not just dopamine, but serotonin, for instance, monoamines in general, that actually, in the right circumstance, given the right input, can be a source of strength, that those cross-fostered young become excellent citizens in their primate communities. They become loved and admired. They experience lots of social grooming and affiliation, and become themselves actually good parents. So it gets back to this idea that with the right intervention, and it doesn't have to be a foster family or an adoption, it can be a teacher who cares, right? Or a child therapist, or some clinician that very early on creates an intervention to break the cycle, that individual, despite any initial harm or injury, whether it be, from a sort of genetic legacy of despair, or trauma, or from an early environmental experience, that individual can grow by leaps and bounds and really accomplish great things.
Okay, great. I think we got maybe one more question. Building from the last question, from my own learning experiences on early childhood trauma, there's a field that studies children that are more resilient and able to succeed. And this may be related to children having a better HPA, hypothalamic pituitary axis. I'm wondering, from what you shared on dopamine, how that interacts with this axis.
Mm-hmm. Yes. So one of the things that, what I didn't get to in the talk is how, despite being a source of strength, enabling us to look upward and outward and imagine new possibilities, building new lives, for instance, in the case of human expansion and migration, that dopamine can actually, when you become excessively dopaminergic, personality-wise, can keep you from attending to the kinds of internal modulation, that here and now process that's mediated by serotonin and oxytocin and the endorphins, for instance. So by learning to self-modulate, to control their stress reactivity, for instance, there are lots of public schools now that are using mindfulness as part of their basic curriculum, introducing that skill of being able to control your own stress reactivity. Children, especially children at risk, can learn when it's appropriate to look sort of upward and outward and seek things that are new and experiment, and when it's important to stay safe and attend to those sort of parapersonal needs and concerns. So And here I think it is dopamine acting in the tuberoinfundibular pathway that may play a role in this ability to shift from, fight or flight, I'm going to try everything and anything, to a more sort of parasympathetic based response to stressors and challenges in the environment.
Great. Well, on behalf of everyone here on the call today, I'd like to thank you very much for this fascinating lecture. It's given us a lot to think about, and it's wonderful I'm such a fan of evolution in general, so I'm really thrilled to be kind of integrating that kind of concept into a lot of things that you're thinking about. And I hope we all get to see you soon in person. And for now, I'll just say thank you on behalf of everyone, and thank you for the great questions as well. So thanks, everyone, and hope to see you soon.
Thank you. It was wonderful meeting lots of you for the first time. And see you in class tomorrow for my students. Bye.
Bye.